Single cell is one of those things that needs to work well every time. And I think once I started to see how robust the 10x data was, it was a no-brainer. It was a no-brainer. We had to switch and incorporate that into our workflows.
The protocols were straightforward, simple, but the most important thing, and having, to be frank, messed around with other types of single-cell technology at the time, was the quality of the data was fantastic, really fantastic.
When we look at the data, we can immediately see the anatomical regions of the brain appearing very clearly. And from there, we find many of the gene markers that are not reported before, but match and expressed specifically to that particular location of the brain. So yeah, it's never ending in terms of having surprised when we see the data.
Layering additional assays that allow us to quantify other types of genomic information at the level of an individual cell, I think ultimately just allows us to increase the depth of the biological questions we can address, and ultimately, speed up the progress to understanding, the fundamentals of cellular biology.
So all of our immune cells in the body come from stem and progenitors in the bone marrow, but we don't exactly know which of the cells makes which cell type. And the clues should be there, but in order to get those clues, you really need to get the genes on a per cell basis. And so when we did, you could clearly see cells that were already primed towards all of these different cell types that we knew about.
It was amazing. So before looking at the 10x Visium data, we generated some of the legacy spatial transcriptomics dataset with a much lower resolution, about 50 to 100 cells per spot. And that dataset were already very impressive. But when we saw Visium data with like 10 times higher in terms of resolution, that really made us think that that's where we can explore the biology.
So single cell approaches for us completely solved that problem. It allowed us to basically say for a different cell type, how does the genetic effect vary in a certain type of T cells or a certain type of B cell. And on top of that, the advances that have happened in the computational space, the analysis of all this single cell data, we're an even more exciting position where we can start addressing how these genetic effects occur in different cell states.
I think it's analogous to having a super telescope for the first time and looking at it, and suddenly there's a part of the universe that opens up that wasn't there previously to the technology you had. And it was that same experience when we first looked at our single cell data.